Good evening. My name is Larry Dubinsky, President and CEO of the Franklin Institute, and welcome to this special speaker series lecture. Really want to thank you for joining us tonight, and it's my pleasure to really welcome each of you, our closest friends and supporters, in this virtual edition of our speaker series. The Franklin Institute has a long history of offering these important conversations on timely issues in science and technology. Tonight, we're grateful to be joined by Dr. Paul Offit, a member of the Institute's Board of Trustees, Director of the Vaccine Education Center at the Children's Hospital of Philadelphia, and a member of FDA's Vaccine Advisory Committee. There's actually a lot more on Paul's bio, but we would be here till tomorrow morning. I know he'll share some of those with you, and many of you may have seen Paul even earlier today on CNN a couple of times. Together with Paul is our host, Dr. Jayatri Das, the Franklin Institute's Chief Bioscientist. Well, tonight we'll learn more about the current race to a vaccine for the coronavirus. Before I turn it over to Dr. Offit and Dr. Das, I do want to thank you for all of your incredible support. Since March, when the Institute's doors closed and when we reopened in July, philanthropy has made the key difference for the Institute. Our loyal donors, our members, our visitors, and friends have sustained us so that we will not only have survived at this difficult time, but we are well positioned as we go forward. Our Franklin at Home programs, including live Facebook sessions on COVID-19, the night sky, environmental issues, our Spark of Science videos, science recipes, at-home hands-on activities, educator workshops, and more have reached literally millions of people. For example, last week we launched a new program for the School District of Philadelphia, and we received an overwhelming response from teachers. Actually, we had to cap the number of applications we received in a 24-hour period, which were almost at 200, twice the number we had originally planned. But we'll continue to do more of this as we go forward because science education is so important. Additionally, our signature youth programs, PACS and STEM Scholars, finished the year virtually but strong, and we've already started this year's PACS and STEM Scholars programs, helping those most underserved students get engaged in science and technology education and a path forward. As we work to inspire the next generation of STEM leaders and build a more scientifically literate society, perhaps now more than ever, I can't thank you enough for your generous philanthropic support. I also have many trustees on this call this evening and want to thank them. Their leadership during these unprecedented times have been amazing. I thank our chair, Don Morell, and our entire board of trustees for their tireless efforts. I do look forward to a time when we can all gather safely at the museum again for these type of programs. But please note the museum is open 5 days a week, Wednesday through Sunday, with safely opened, and has really been a model, both here in Philadelphia and around the nation, of how you can reopen safely and engage people in some of the most important topics of the day around science. Until then, I'm pleased to introduce Dr. Jayatri Das and Dr. Paul Offit as they present the race to a vaccine.
Larry, thank you so much. And as we get started, I want to first, of course, thank Paul Offit for being with us here today. Paul, you were mentioning that you've never been busier, and for a virologist, that's sort of an alarming situation because it means that things are really bad. And we're in this situation where, on one hand, science is advancing at a global scale in a way that we've never really seen it galvanized before. And there's a lot of excitement in terms of the work that's being done. Yet, on the other hand, that very speed of the science is something that's giving a lot of people pause. And of course, with a vaccine, it's great to have a vaccine, but unless people trust it and take it, it doesn't do us any good. And so the conversations like this, and for all the tireless work that you're doing out in the media, Paul, the important piece is to help people understand, the process so that we can be confident in the outcome of that process. And all of us play a role in that. We're each science communicators in our own way to our friends and to our family. And so what I hope that by the end of this hour, after we have a chance to listen and learn from Paul, that all of us can share that knowledge, that we're all, that those science communicators, trusted communicators to our friends and family, and you can help everybody understand this process and what to watch out for. So Paul, thank you for everything that you're doing and for taking the time to be here with us today. So as we get started, Paul, give us, that 2-sentence summary of what a vaccine does for an infectious disease.
Right. So what a vaccine does is it induces the immune response that is a consequence of natural infection without having you pay the price of natural infection. So when you're trying to make a vaccine, the most important thing you want to see is you want to see that natural infection can protect against reinfection, or at least re-exposure to the virus. That's the most important thing. That's not true of all things. I mean, for example, you can get strep throat again and again and again. That's a bad sign. There's an example where natural infection doesn't protect you against disease associated with re-exposure, and that's why we don't have a strep throat vaccine. Or gonorrhea is a disease you can get again and again and again, and having one infection doesn't protect you from the next one. So I think all the evidence so far is that SARS-CoV-2 virus, the virus that causes COVID-19, does induce an immune response that appears to protect. Because although you hear the sporadic cases, and there was one just reported in Lancet Infectious Diseases of someone who had a moderate to severe disease, clearly got better, and then a second time also had a moderate to severe disease, those are reportably rare. So I think all the signs are good that we can make a vaccine.
And that's the most important baseline. So many of our questions as we go forward in this conversation are pulled from the enormous list of questions that all of you sent in advance. So we'll try and get to as many of them as we can work into our conversation. And if others come up, please go ahead and put them in the Q&A. You can also, chat with each other or share your expertise in the chat. We know many of you have some experience in this field as well, and we'd love to hear from you. But please go ahead and put your questions in that Q&A so we can work them into our conversation. So I wanted to start out with kind of a high-level overview of the science behind the different vaccines. So there are different ways to create a vaccine, and we're hearing these, we're hearing this idea of an RNA vaccine, we've heard of a vector vaccine. Can you explain like the 3 or 4 top contenders and the science behind them?
Sure. So the good news is that we believe we know the part of the virus that we're interested in. It's that spike protein. It's the protein that emanates from the surface of the virus that makes it look like a crown. That's why it gets the name corona. That protein is responsible for binding the virus to cells. So if you can make an antibody response to that protein, then you can prevent the virus from binding to cells, you can prevent the virus from infecting cells, or said another way, you can prevent the virus from infecting you. We also know the gene that codes for that protein. We know the virus gene that codes for that protein. So all the early attempts to make a vaccine are based on that concept. They're the so-called genetic plug and play vaccines. So what you do is you take that gene and then you can put it into a variety of scenarios. But the goal then is to inoculate you in some form with that gene. And so that then your body makes the coronavirus spike protein, and then your body makes antibodies to the spike protein. Those are really all the initial strategies, and they have names like mRNA, DNA, replication defective, simian adenovirus, replication defective, human adenovirus, vectored vaccines, so-called replication competent vectored vaccines. They all have different and confusing names, but it all boils down to this. You inoculate the person in some form with that small gene that codes for the spike protein, then it induces you to make the spike protein. That's it. Now, the reason those were chosen was they were the easiest to construct. I mean, you just plug in the gene, hence the plug and play phrase. Not necessarily, and because they're very easy to scale up. So the initial strategies were chosen because they're the easiest to construct and the easiest to scale up. And one could reasonably say, well, does that necessarily make them the best? No, it doesn't necessarily make them the best, it makes them the fastest. And I think over the next couple of years, we're going to see what emerges in terms of what are the best vaccines. But I think that's why these are going to be the first vaccines.
I think you've hit on a couple of important points there that came up in the questions. First that, all of these different types of vaccines that we're hearing about essentially are still the same strategy in terms of what part of the virus is being presented to your body. And second of all, the first ones are the fastest. And we're at this place because we're essentially standing on the shoulders of vaccines that have been developed previously.
That's an Isaac Newton quote, right? I see farther because I stand on the shoulders of those who preceded me, right?
So yeah, you got to work in all the scientists here. But one of the questions has been like, you've said that the vaccine that you worked on for the rotavirus took decades. How can we get from a novel virus to a vaccine at this stage of testing so quickly, just from a scientific perspective?
Right. So great question. So, I mean, the typical time it takes to make a vaccine is 15 to 20 years, roughly, on average. If you look at the last 20 vaccines, that's about right. The vaccine that I was fortunate enough to work on at Children's Hospital of Philadelphia, the rotavirus vaccine, took 26 years. That's not that abnormal. So how is it that we can have a virus, this SARS-CoV-2 virus, in hand in January and reasonably conclude that we will probably have a vaccine early next year, in a year and a quarter? The reason is, well, I guess, just take a moment to talk about how vaccines are developed. I mean, you first you do preclinical trials in experimental animals. So you make sure that so-called proof of concept studies. Does this work to protect an experimental animal from disease? Then you go to phase 1 studies, which are basically small dose ranging trials. Then you go to phase 2 trials, phase 2 studies. So now you know the strategy you're going to use, you know the dose you're going to use. Just want to make sure it's safe and continues to produce an immune response. Then you do phase 3 studies, which are these big tens of thousands of people, prospective controlled studies where you're going to see whether your vaccine works, and then you mass produce it. What's happened here is the government has basically taken the risk out of it for pharmaceutical companies. They've said, we'll pay for phase 3 trials, which cost hundreds of millions of dollars each. We'll pay for mass production. We will mass produce this vaccine before we even know whether it works, before we even know whether it's safe. That costs also hundreds of millions of dollars. No company would ever do that. That's why it is that it's so fast. Now, and you can see why that would be worrisome to people. I mean, phrases like warp speed, the race for a vaccine, who are the finalists, does not exactly make people feel comfortable that things are being developed the right way. I think people would reasonably say, are there critical phases that are being skipped, or worse, are there safety guidelines that are being ignored? No, the answer is no. As long as we let the phase 3 trials proceed the way they're proceeding. I mean, the Pfizer's trial is a 30,000 person trial. Johnson & Johnson's trial is a 60,000 person trial. Moderna's is 30,000, Pfizer's is 44,000. So, I mean, that's about right. I mean, the human papillomavirus vaccine trial was a 30,000 person trial. Conjugate pneumococcal vaccine was 35,000. So as long as we do that trial and let it proceed to completion, I think that that's what you want to make sure you see. So I think I wouldn't be worried about this process.
So I think that's an important point, that that we're building on science that's worked before. And it's the resources that we're marshaling towards supporting this effort that is unprecedented. But that doesn't necessarily have to mean that we're compromising the integrity of the process. So let's get into some more details of the process, because there are a lot of questions that we got about understanding how vaccines are tested for efficacy and safety. So let's start with talking about some of the questions around phase 3 trials. So one question that several people asked, knowing that even with tens of thousands of people enrolled in these phase 3 trials, how much diversity in age, in background are being included in these trials?
Right. So actually on the NIH's National Institutes of Health so-called active group that was put together by Francis Collins. So we were the ones who actually set up for how we thought these trials should be designed. We felt it was important to make it look like the American population. I mean, if you're going to recommend a vaccine for people who are over 65 years of age because they're at a special risk, you have to make sure you have a representative number of people in that group who are over 65, so that you can say this is effective not only in the general group, which is for the most part going to be healthy young people, but it is effective and safe in this group because we've tested this many thousands of people. because there are differences, right? I mean, people who are over 65 make a very good immune response to the shingles vaccine, but they don't make a very good immune response to the flu vaccine. So there are differences in age in terms of how you respond. So you need to know that you can say that for those people. And you also want to make sure, obviously, you have the adequate representation of minorities, of racial and ethnic groups. You have representative Black population, Latinx population, not because there's likely to be differences in their ability to make an immune response or differences in their ability to be protected, but you have to when you're going to go to folks and say, look, you're represented in these trials, you're here in these trials. I think that is going to be more convincing than just saying we tested it in a group of people who in no sense look like you. So Pfizer actually slowed their trial at one point because they were not getting enough in the way of ethnic and racial minorities. So because it's clearly something we all agree has to be done, and I think it will be done.
And I've read about some great partnerships with medical schools at historically Black colleges and universities to really do that outreach and ensure the diversity that we need. Now, you mentioned older populations. One population that isn't yet, these vaccines aren't yet being tested on our kids. What's the prognosis for when we might start seeing pediatric populations being tested?
Right, so children so all the trials that are currently ongoing are in people over 18 years of age. I mean, Pfizer just recently, when they expanded their trial, dropped it down to 16 years of age, but for the most part, over 18. The reason for that is that people less than 21 years of age make up about 26% of the US population, but they only account for 0.08% of the deaths. Actually, more children died of influenza last year than have died of COVID-19. So they're not a priority group. That said, I think ultimately we are going to have a vaccine for children, and the only way we're going to have a vaccine for children is if we test it in children. We're not going to recommend a vaccine for children that hasn't been tested. The question is, how can that be done? And I think it will be done probably in a placebo-controlled way. But the good news for children is we'll have a lot more information, I think, by the time we finally get to children about these other vaccines, what their strengths and weaknesses are, so that we can make a better pick for children.
And certainly that's important because we know that, while most children, at least on the younger end, don't suffer as severe symptoms as older adults, they are still, they can still transmit the disease to others. And so it's important to break that chain of transmission with kids as much as others. So what are these trials looking for when they're looking for evidence of efficacy?
Right, and that will be a big discussion next week at the FDA's Vaccine Advisory Committee meeting. The tension here is, well, I actually should pull out this little graph that I was sent by the head of the Clinical Trials Subcommittee of the NIH group. See, can everybody see this? But, the tension is the so-called interim analysis. There is a push by the companies to want to do an interim analysis, meaning to say that let's stop when we have, say, 35 cases of disease and see where we are. And let's say there's 20 cases of disease or 30 cases of disease in the placebo group and 5 cases in the vaccine group. And then we can say, look, it looks like it's working. Let's get it out there. But most people who are on that committee are going to want to see at least 150 cases of disease because statistically you want to have something that is robust, that is significant, that has very tight so-called confidence intervals, so you can say with a high degree of confidence that something is effective. And you also want a representative number of cases of severe disease so that you can say, yes, I think it's likely that this vaccine not only works, but keeps you from dying. And so that's going to be the tension at this meeting. On the one hand, there's a disease out there that's killing hundreds of people a day, sometimes as much as 1,000 people a day. On the other hand, remember, most of these vaccines are going to be given to healthy young people. And so you want to make sure, first of all, that it's safe, and secondly, that it's clearly effective. Because if it's not, if you have a vaccine that's like 30 or 40% effective, and you put it out there, and there are people who get the vaccine, now they're thinking, great, I'm good, I'm immune, these viruses are going to bounce right off me, I'm going to take off my mask, I'm going to engage in high-risk activity, and then they get sick, you're going to hear about it, and you're going to lose confidence in this vaccine. And there's already a fragile confidence for vaccines in this country, and we can't afford to make that kind of mistake at the beginning. You only get one chance to make a first impression, and if you scare people off vaccines, if they think, that stuff doesn't work, it's a problem, because there's 2 ways out of this pandemic, and we're going to need both. One is hygienic measures, right? Masking, social distancing, which is the most powerful. That is the most powerful thing you can do. If you ask me, when I'm walking down the wards at Children's Hospital of Philadelphia, which I will be doing next week, and I have three choices. I can either social distance from, let's say I'm walking into the room of someone who has SARS-CoV-2. I have three choices. I can either stand 6 feet away from that person, or if I can't stand 6 feet away from that person, I can either wear a mask or have a vaccine. Which would I choose? Number one, stand 6 feet away. That's the most powerful thing I could do. Number two, if I can't stand 6 feet away, wear a mask. That's going to be far better than a vaccine. And I think people are thinking of vaccines in the wrong way when they think of it as this magic powder that we're about to spread over the country and it's all going to magically go away. It doesn't work that way. I mean, we need both. Even if you've gotten a vaccine, let's say the vaccine's 75% effective, which I think we'd all be happy with. Even if you've gotten a vaccine that's 75% effective, which is good, right? 75% effective against moderate to severe disease. That means 1 out of 4 people could still get moderate to severe disease when exposed. And you might be that 1 out of 4 people, so you should still wear a mask. And I think that's going to be a hard sell, considering it's hard enough to get people to wear masks now without a vaccine.
Yeah, I've been seeing an analogy to Swiss cheese lately, that you look at any one of these interventions as a slice of Swiss cheese, that it's got holes in it, it's not perfect, but you start adding them up together, and when you get the whole stack of slices together, they compensate for each other. And so altogether, we have to keep this multi-pronged approach to prevention alive, and we'll come back to that at the end when we think about what's the path forward. But as we think about a couple more questions around the efficacy and the safety of the vaccine, the FDA has said that 50%, I believe, is 50% efficacy is the standard for considering approval. Obviously better, more, a higher percentage is better, but what's your perspective on how to communicate why a 50% effective vaccine is better than nothing?
Right. So when the NIH active group that was put together by Francis Collins, director of the NIH, was trying to figure out what was acceptable and not, the statistician from the University of Washington set as a minimum 50% efficacy with a lower bound of no worse than 30% and an upper bound of, or was it 50%, 30% lower bound and then about 60% upper bound. That was just an exercise. That was an exercise to sort of play out the statistics. All of us believe that we can do at least 60% efficacy and probably higher. So I mean, I think the reason probably 50% was set is that the influenza vaccine is about 50% effective. So, and that is a licensed marketable vaccine. It's interesting how this is going to play out. Typically vaccines are licensed. They're licensed by the Food and Drug Administration, and then they're recommended by the CDC. And we all wait for that recommendation. Insurance companies wait. Pediatricians wait, adults and internists wait. We wait for that ACIP recommendation. That's going to be true here too. The difference is that it's probably not going to be a typical licensure process. It's probably going to be emergency use authorization, which I think frankly it shouldn't be, because emergency use authorization really is, you can't have an emergency use authorization for the entire population. I mean, it really goes against the sort of whole notion of emergency use. And the term authorization is not right. It's really a permission is what it is. It's a permission to use that product is what it is. And so I worry about this a little bit, that the standards may be looser than they need to be, but I'm hoping that they won't be. And I do want to sit on a committee where at least I get a vote.
And we'll come back to that as well,
Right.
About the approval process. But one last question that I want to get to in terms of the science of the vaccine is related to a question about the mutation rate of the virus. So what do we know so far about how quickly the genetic sequence of the SARS-CoV-2 virus is changing, and how might that affect the strategy for developing a vaccine?
Okay, so it is a so-called single-stranded RNA virus, like flu. Flu is also a single-stranded RNA virus. And so therefore, like all single-stranded RNA viruses, it mutates. It mutates every time it reproduces itself. The question is, does it functionally mutate? Meaning, does it mutate so that it becomes more virulent or less virulent? Does it mutate so that it becomes more contagious or less contagious? Most importantly, does it mutate away from the vaccine? Does it mutate so much so that that spike protein changes so much that now antibodies that were induced by the vaccine don't kill that virus anymore? To date, I think you can say, and I should also point out measles is a single-stranded RNA virus. So is mumps, so is German measles. Now those three viruses also mutate all the time, but they don't do what flu does. So that's the question. Is this virus going to be more like flu, where it mutates so much every year that you need to get that natural infection or immunization doesn't protect you, and so you need to get a yearly vaccine? Or is it mutate like measles, which has been a stable, basically one serotype virus for its entire existence? I think the answer is it's going to be more like measles. I don't think it's going to, although it does mutate, and it does appear that there's at least a mutation that's associated with greater contagiousness. There's no evidence that it mutates to become more or less virulent, meaning dangerous, and there's no evidence that it mutates away from the vaccine. So I don't think that's going to happen. Now, I don't know how long immunity is going to last, but I'm going to predict, hopefully, this is being recorded, bummer. So just don't hold me to I'm willing to make any predictions, just don't hold me to them. But I really don't.
Any prediction may change by next week anyway.
I don't think it's going to require a yearly vaccine because it's like flu. And I imagine the vaccine would induce protection for years, would be my sense of it.
And I think, one thing to understand about, genetics in general is that all genetic sequences change. It's a question of, whether those changes are happening in the parts of the virus that are involved in that infection process.
Right, there's a change function, exactly right.
Right, so we will see changes in the sequence overall, but many of those changes just won't have an effect in the impact of the virus. So I want to take a quick detour here because we've had some questions about the emphasis on developing vaccines versus a couple of the therapeutic approaches that have been in the news lately. And of course, the difference here is that therapeutics are treatments that can help reduce the severity of disease in somebody who's already been infected, whereas the vaccine is more of a preventative approach. So one that we've been hearing a lot about is monoclonal antibodies, and Regeneron in particular is a company that's developed a cocktail of monoclonal antibodies that was most famously recently used to treat President Trump. Can you talk about the difference between monoclonal antibodies delivered as a therapeutic treatment versus how a vaccine elicits an antibody response?
Right. So if you're infected with the natural virus, SARS-CoV-2, you make so-called polyclonal antibodies, which is to say, so here's a virus that's made up of 4 proteins. You will make an antibody response to all 4 of those proteins, and you'll make an antibody response to a number of individual segments of each of those proteins. That's a polyclonal response. That's what your body does. Monoclonal antibodies are synthetically made in the laboratory that are directed against one part of one protein. In the case of the Regeneron product or the Lilly product, it's a monoclonal antibody directed against one domain on the spike protein, the so-called receptor binding domain, which is the key part for neutralizing the virus. And the Regeneron product has 2 monoclonal antibodies, that's the cocktail part, both directed against that receptor binding domain. In terms of the critical questions, does it work? I mean, the problem, it's really easy to kill bacteria because they reproduce themselves outside of cells. Viruses reproduce themselves inside of cells. So that's always made therapy hard. It's hard to sort of kill the virus without hurting the cell. So the question is, do monoclonal antibodies or polyclonal antibodies work? I will say that historically, antibodies have a checkered history in the treatment of infectious diseases. I mean, if you're infected with measles, for example, you're given a measles-specific immune globulin, if given in the first 48 hours of exposure, will prevent you from getting disease. That's also true for rabies. It's also true for hepatitis A virus. It's also true for chickenpox. If you're infected with chickenpox and you're given essentially polyclonal antibodies, you're protected from getting the disease as long as you give it right at the beginning. If you wait a few days, it's too late. Even though you don't have symptoms yet, it's still too late. See, that makes you think that antibodies are not going to treat a disease if in those 4 instances that they don't work to treat the disease. Now, the best news for monoclonal antibodies that they might work was Ebola. That was the first example where monoclonal antibodies clearly could treat a disease. Because the way viruses work is when you're infected with a virus, it enters your nose and throat and starts to reproduce itself hundreds, thousands, tens of thousands of times. You know, it comes up and then your body starts to make an immune response. That's when you get symptoms. It's the immune response that causes the symptoms, not really the virus per se. As the immune response comes up and the symptoms then come up, the virus replication goes down and down and down. So by the time you're really floridly symptomatic, virus replication is not such an important part of the disease process. That's why antivirals like remdesivir are not exactly wonder drugs. I mean, they save a few days of disease, but they don't save you from, they don't save you from dying. That also may be true with Regeneron. And I think that despite President Trump's claim that this clearly cured him, first of all, he was also treated with oxygen, dexamethasone, and remdesivir. So all three of those could have contributed to his doing better. He also was actually fairly early in his illness. But he claimed it was Regeneron, and so it must be the Regeneron. I'm just kidding. So I think what you need is a study. And what worries me is if Regeneron is able to convince the FDA to have an EUA approval, it's going to kill any studies. And we really need to know whether it works to treat disease. We need to know that because it may well not treat it at all. We need to find that out.
And we've seen that with some of the other drugs that have been proposed as therapeutics. And I think you explained it well in terms of thinking about the course of the disease, that there are some therapeutics that are targeted at reducing the replication of the virus in the early stages. And then there are cases as the disease gets more severe where your immune system actually overshoots. And that's a problem in and of itself. And there are other drugs that are then used to treat that. We have a couple questions about how, why we're seeing now fewer deaths and ICU cases even as infections continue to rise. Is that because we have a better understanding of the progression of the disease and the drugs available?
Yeah, I think, yes, a number of things. I think we've learned that we shouldn't be so quick to intubate patients, that it's really more of an oxygenation problem, meaning getting a decent percentage of your blood cells carrying oxygen, and then a ventilation problem, meaning getting CO2 out of your blood. So we've been, the trick is getting oxygen into the blood much more so than getting CO2 out of the blood. So I think we were quick to put people in ventilators where all we really needed to do was put them on high dose oxygen. I think that high flow oxygen. The second thing I think is that dexamethasone. I think that it's always a balance, right? When your body makes an immune response to kill the virus, it also hurts you. I mean, in many ways, it's your immune response that kills you. And so there's always that balance between suppressing the immune response and because when you suppress the immune response, yes, you make symptoms better, but you also make it more difficult to rid yourself of the virus. I mean, the antibodies that you make, the so-called killer T cells, which kill virus-infected cells, get also suppressed by that dexamethasone. But I think that's the lesson here, is that dexamethasone matters. The other thing that we've learned, because this virus is so surprising, is it causes vasculitis. It causes inflammation of blood vessels. That's the problem. I mean, when I was on service a couple of weeks ago at Children's Hospital Philadelphia, we had a teenager in the hospital. Her chest x-ray, which was a pneumonia, did not look like any viral pneumonia I've ever seen before. It looked more like lupus. It looked like a rheumatologic disease, a vascular disease. That's what it looked like. So I think we've learned also to give anticoagulants, because people essentially have this hyperclotting syndrome. So I think that's all been learned over time. Initially, we were just treating this like a typical viral pneumonia, and it's anything but that. So I think we've gotten better at it.
And that's good news, certainly, for for keeping everybody healthier and back to recovery faster. Now let's switch gears into the approval process for vaccines, and certainly this is applicable beyond vaccines. We've seen some of these other drugs go through trials and go through that FDA process. So maybe we can break down this in a more generic way. But when do we expect to see that, reaching those targets of 150, 160 patients with COVID to be able to tell whether these vaccines are working? What's the timeline?
I'm glad you've asked. If you look at the 5 vaccines currently in the warp speed bucket, as it stands, Moderna's messenger RNA vaccine, we should have 150 by mid-January. For AstraZeneca's vaccine, and now we've just had a break. So it's it's back on, it's off clinical hold in Europe, but it's still on clinical hold here. It'd probably be sometime around the end of March. For Janssen's vaccine, which is another, that's a single dose vaccine, that now also got put on clinical hold. So we'll see how that plays out. But as it currently stands, about mid-February. And then Novavax and Sanofi, actually, they have purified protein vaccines. So there you actually give the protein and then the person makes the antibodies to the protein. Those are going to be April or May to get to 150. And so that's going to be the fight. I think at the FDA Vaccine Advisory Committee meeting, there's going to be the 150 group, and then there's going to be the 35 group. And we'll see sort of how that plays out. I'm going to be a member of the 150.
So I want to explain kind of quickly, where those numbers come from, because they kind of seem arbitrary, but in fact they're based on statistical calculations of the statistical power that you have to detect efficacy of the vaccine. They're based on the frequency at which your enrolled population of participants is likely to encounter the virus, in their everyday life after they get the experimental vaccine. So why are these numbers what they are?
Right. So it's I'm not a statistician, but I'll try and explain it as I understand it, which is to say simply and probably wrong. But here's how I understand it. I mean, so let's suppose you were doing a trial and you had then you had 20,000 people that were getting vaccine and 20,000 people that got placebo. And now you're waiting for people to get sick, and you find that 5 people get sick in the placebo group and 2 people get sick in the vaccine group. Well, that's more, right? There's more in the placebo group and less in the vaccine group. So that looks good. Looks like it's trending toward it could work. But those numbers are very small. And if you just continue along over the next few weeks, it may be 10 and 10. So you haven't really hedged your bets much. So now let's suppose it's 35 cases in the placebo group and 10 in the vaccine group. That's still pretty small. And if you go along, now down the road, it could be 50 and 50. But if you did, let's say you have 150 and you have 140 cases in your placebo group and 10 cases in your vaccine group, now you're getting more and more sure. I mean, the whole point of doing trials, whether you go from phase 1 to phase 2 to phase 3, is to reduce uncertainty. That's what you're trying to do, reduce uncertainty. You're not trying to eliminate uncertainty. There is no eliminating uncertainty. There's always a certain amount of uncertainty. And that'll be true here too. I mean, even if we do a trial of 40,000 people, say 20 and 20,000, and we see that it's 75% effective, and the numbers are really good in terms of placebo and vaccine, you still have only tested it in 20,000 people, the vaccine. So, and that's not 20 million people. So you can rule out a relatively uncommon side effect problem, but you haven't ruled out a rare side effect problem, but there are systems in place to detect that. You can also only say that it's effective for however long you've tested it, 4 months, 6 months. You don't even know whether it's effective for a year. That too, you won't find out till after the vaccine is already out there. So it's always a series of steps of trying to mitigate risk, lessen uncertainty. But you always have to have systems in place to continue to try and lessen uncertainty over time. I mean, you can say for a vaccine like the measles vaccine, which has been around for almost 60 years, that you have pretty much feel pretty confident that vaccine is safe and effective. That there's not going to be any surprises, but there's always surprises. The first vaccines are often not the last vaccines, and there's many examples of that.
And so, you're saying that we have to make sure that there are systems in place, as safety nets for these. And certainly this process of the clinical hold when there's an adverse effect is part of that safety net. Can you explain that process of what happens when, a serious side effect is discovered?
Right. So the overseeing every vaccine trial is something called the Data Safety Monitoring Board. These are people who are not associated with the government. They're not associated with a pharmaceutical company. They are academics, independent academics. They are charged with, if there's ever any serious event that occurs, let's suppose somebody has symptoms of multiple sclerosis, or somebody has something called transverse myelitis, which is what happened with the AstraZeneca trial, which is to say inflammation of a segment of the spinal cord that then causes symptoms associated with spinal cord injury, which can be difficulty moving, difficulty in sensation, et cetera. And it occurs within a week of getting the vaccine. Could the vaccine have done it? Remember, the vaccine is designed to prevent SARS-CoV-2, That's all it's designed to prevent. It doesn't prevent everything else that happens in life. So you're going to always deal with these coincidental associations. The question is it a causal association? So the Data Safety Monitoring Board is instructed that whenever there's an apparently serious adverse event that's occurred within a certain period of time of getting the vaccine, stop. And then take a very careful look at that patient, take a look at other patients who have gotten the vaccine to make sure that, you look at their safety records and make sure that there's nothing similar going on. And then you can take it off this clinical hold. And these have been clinical holds, pauses. I mean, the Johnson product, Johnson & Johnson product, that was essentially a clinical pause. So it's the Data Safety Monitoring Board that did it, not the regulatory agency. If it starts to look bad, if the regulatory agency stops, that's when you wonder whether or not it might be bad. So it's good. It's what you want. You want to err on the side of caution. The problem is we're so desperate for a vaccine that although these clinical holds or pauses occur all the time, everybody's panicking every time one occurs. I mean, I was interviewed on CNN by Brian Todd today on CNN, and he wanted me to say that. He wanted me to say this was bad news. But I don't think it is bad news. So.
Right. And I think, having that independent monitoring board is so important to ensure that there's an external review of the process, that it's not just all people who have, different stakes in the process. So I think that's a really important piece to communicate.
Can I say one other thing, Jayatri?
Yeah.
It's the frustration of the clinical hold is that when you put something on clinical hold, everybody knows about it, right? Because the investigators all just got told to stop. Therefore, the public knows about it. Therefore, the press knows about it. What you don't know about is exactly what that case was. What are the details of that case? Technically, it's confidential. So you can't really know what that case was. I mean, he called me actually, the head of the Data Safety Monitoring Board in the United Kingdom when the AstraZeneca product was put on hold. So we had a conversation. I asked him, so tell me the story. I mean, what exactly are the details of this case? I can't tell you that. It's confidential. It's like I go to jail if I tell you. I cannot tell you that. So what happens? So therefore, we shouldn't know anything. What ends up happening, though, sometimes is that the chief executive officer, or the chief financial officer, or chief medical officer ends up doing a meeting with the shareholders and then gives some information. And then that goes to the New York Times or whomever. And so then you get it's transverse myelitis or it's multiple sclerosis, so you get a little bit of information. So my theory on these things is if it's confidential, then don't say anything. Don't just leak little bits of information. It's all very frustrating and people get angry that people aren't being transparent. But frankly, they could be more transparent. I think the whole thing about confidentiality is you can't give information that would in any way identify who that patient was. So where this happened, what county it happened in, for example, or more specific details of age of the person or gender of the person. Don't do that, but you can be more specific actually about the disease itself. They aren't. They just don't give you that information. So it's all very frustrating.
And to your point about transparency, I mean, that's certainly a part of the approval process that I think is essential to communicate to people. So we know what those milestones are, that they're being met. And to that end, a number of the companies have actually published their protocols so that people can understand what those milestones are. That being said, there are obviously questions about the political pressure that's being applied. And you're involved in the decision-making process. Can you give us a summary of what those levels of approval have to be before a vaccine is approved for widespread use? And what confidence can we have that politics is not going to drive that?
Well, I would have said 5 years ago, you could have tremendous confidence that politics wouldn't drive it. That hasn't been the recent story. I mean, clearly there's been a politicization of science. No matter what what party you support, there is just no denying the fact that that when the Environmental Protection Agency has its arm twisted to take the phrase climate change off its website, that the National Oceanographic and Atmospheric Administration is asked to influence the National Weather Service to say that Hurricane Dorian actually did enter Alabama when it didn't, to support the president. When the FDA is pushed to approve or at least permit the use of hydroxychloroquine, which doesn't work to either treat or prevent the disease and can cause heart toxicities, arrhythmias, or convalescent plasma, which also has never been shown to work. And there are a couple of reasons, there are sort of technical reasons why it actually may hurt. You can't help but worry that the FDA has been politicized, that the FDA now sees its role as just serving at the whim of the president and not doing its job, which is to protect the American public from products made by pharmaceutical companies that are unsafe or ineffective. That's what you can't help but feel that. And that's why people get scared. And now, as an example of that, you have just chaos. You have, for example, the 5 states that have said, we're not going to listen to what the FDA says. We are going to form our own expert panels to decide whether we're going to get vaccines. You've had the pharmaceutical industry band together and have whatever, 5 or 6 pharmaceutical companies wrote a letter saying you can trust us, because they sense that people don't trust the FDA. So that's what they're saying. They're saying you can trust us because obviously you don't trust the FDA to regulate us. I mean, you have the president saying, look, you can trust pharmaceutical companies. Well, I mean, that sort of belies 100 years of drug regulation in this country. I mean, take your pick. You know, any of the major drug regulations, Biologics Control Act, the Food, Drug, and Cosmetic Act of 1938, the Kefauver-Harris Amendment, all of them, the Cutter Incident, which gave birth to the Division of Biological Standards, every one of those was associated with a major pharmaceutical company disaster. So
let me rein you back in, Paul, because I'm curious. You know, you have a seat at the table. So where do you see your role, like your committee's role in the process in helping bring transparency to this approval process?
Right. So I think so. How do we heal this? I mean, how do we heal what I think is a fractured relationship with the FDA? I think people don't trust the FDA right now. Kamala Harris basically said that. She said, I'm not going to trust what the president recommends. Well, what the president in theory is going to recommend is what the FDA recommends. I would like to think is what the FDA is going to recommend. So how do you heal that process? I think, first of all, there is no reining me in, Jayatri, but nice try. I think we just have to be transparent. There's some good signs lately. I think that when the FDA said, we want 2 months of safety follow-up after the second dose or after whatever the final dose is, there was pushback from the administration, because that became very clear. If we were going to do that, we were not going to have a vaccine by election day. And so the administration said, I'm going to bypass the FDA. Well, what the FDA did was they basically put out a document saying, this is what we're going to do. I mean, for our committee, we got a document that says, this is what you have to do. 2 months of safety follow-up, and then the administration backed down. So I think the FDA is finally standing up for itself. Good. Secondly, I think that when we look at those data, I think we have to try and explain to the public in a transparent way what we know and what we don't know. But the FDA is just the first part of the process. The more important part of the process is the ACIP, the Advisory Committee for Immunization Practices to the CDC. The FDA licenses products. The FDA says to pharmaceutical companies, you can now sell this product. But it's really the CDC that says, here's who I would recommend it for. Insurance companies usually wait to see what the CDC says. Doctors usually wait to see what the CDC says. So I think, again, the CDC will independently review these data and will come up with their recommendations for who should get which vaccine. That's what I would wait for.
So that there are these layers in place, that it's not, one stroke of a pen and it's out there, but rather there are layers of these recommendations that are in place as safeguards. So as we think about the day when a vaccine does get to that point, there are still lots of questions about how the vaccine is scaled up for production and how it's distributed. And certainly one of the big questions on everybody's mind is, how does this get rolled out? What's the time frame for when we might see the vaccine, being available for folks, even outside of healthcare settings?
Right, so there are 2 groups that decide who should get it first. One is, again, the Advisory Committee for Immunization Practices to the CDC. The other group is the National Academy of Medicine. They basically agree. So the 4 groups that are important are: 1, healthcare workers, because they're on the front lines. 2 is other essential workers who are not healthcare workers. So people in education, transportation, mass transit, food services, law enforcement, et cetera. The 3rd group are people with certain high-risk medical conditions like chronic lung disease, chronic heart disease, obesity. And then the 4th group are people who are older, people who are greater than or equal to 65. If you add up all the numbers for how many people that adds up to, it's about half of the adult population. So it's a lot of people to get what is likely to be a 2-dose vaccine. And the interval is about a month apart for each of those doses. So it's going to be now, we're going to be the points of care, not just doctors' offices. Obviously, they couldn't handle all this. It's going to have to be large chain pharmacies and universities and colleges and hospitals, et cetera. And you're going to have to identify who those people are to make sure they really fall into the high risk group. And you have to make sure they come back, a month later to get that second dose. So it's going to be hard to do all this. I think it's going to have to be states that take the lead on this, not the federal government.
So one other question that in terms of what we can look for is how are we going to be able to track long-term side effects of the vaccine? What kind of systems are in place, as well as what are the long-term impacts of just COVID infection?
Right. So there certainly are systems in place to determine whether any vaccines has a rare side effect. So when vaccine starts to roll out in the population, there's something called the Vaccine Safety Data Link, which are linked computerized medical health records for about 24 million Americans. There's also something called the PRISM, P-R-I-S-M program, which is linked computerized insurance claims. So you'll know sort of who got the vaccine, who didn't, in millions of people, and then you'll be able to see whether there's a very, very rare side effect. In terms of what the long-term effects are of COVID-19, I mean, this is one weird virus. In my experience as a virologist, I've never seen a virus like this. It's a virus that doesn't enter the bloodstream. I mean, if you look at viruses like measles, they enter the bloodstream, chickenpox enters the bloodstream. This virus never enters the bloodstream, yet nonetheless, it damages the lining of blood vessels and causes so-called inflammation of the blood vessel or vasculitis, which means that any organ that has a blood supply, which is every organ, can be affected. So you can have strokes, heart attacks, kidney disease, liver disease. When people say don't fear this virus, fear this virus. I mean, not just because it can kill you. A flu can kill you, but flu either kills you or it doesn't. It doesn't do what this virus does, which is cause long-term problems. It's a scary virus. Never seen anything like it before. It's a bat coronavirus.
Right. I think that, and I think that's a middle ground that we don't always talk about. That either you're not infected yet, or we talk about the deaths, but those who are infected have this range of symptoms that we're still trying to understand, and that'll take some time. So in our last few minutes, I want to come back to just placing the role of the vaccine in that broader context of public health. We talked about the fact that it's going to take a while for the vaccine to roll out, but what does that mean in terms of our own behavior? How long are we looking at continuing to distance and mask and all of those other slices of Swiss cheese that are helping us, stop transmission?
Right. So another way to ask that question is, what percentage of the population would need to be immunized in order to stop transmission, to get the so-called contagiousness index to less than 1? It depends on two factors. One is the contagiousness of the vaccine, the virus, and two is the efficacy of the vaccine. If you have a vaccine that's about 75% effective, which I think is doable, you would have to immunize about 2/3 of the American population to stop spread, which I think is also doable. Most of the vaccines that we have are given, certainly our childhood vaccines are given in far greater percentages than that. Even the flu vaccine is up in the sort of 50 to 60% range. So I think, and people generally, don't get the flu vaccine often because they're thinking, God, I got to get this every year. It's too much. I don't feel like it. But this, I think people will be more motivated to get this vaccine when it comes out. So I think about 2/3 of the American population. But again, I just really want to emphasize that hygienic measures are still part of this story. You can't have one and not the other. Not for a while. Until we get on top of this, we're also going to have to continue to do hygienic measures. So I think it's possible that in the next couple of years we will get on top of this. I do think that will happen, but we have to be much better at the hygienic measures than we've been. I mean, you look at these rallies where people are maskless and they're hugging each other and giving high fives. It is hard to watch. It's like this denial that we're the only country that does this. We're the only country that has managed to make hygienic measures political. I don't know how we did it, but it is hard to watch.
So one question I want to clarify before we wrap up here is, again, that the vaccine is a preventative measure. It's meant to prevent you from getting infected, versus there are other therapeutics that are, that we're learning more and more about, that can be used to treat people once they've already been infected. And so that vaccine is going to just continue to be part of our toolkit. That you don't get, it's important for everybody to get the vaccine so fewer people actually get infected in the first place. Now, that being said, what's the prognosis for schools?
I think in some ways we could go to school now. I really do. I just think in areas where obviously the percent positivity is very high, no. But I do think that there are other countries who have been able to go back to school using social distancing and hygienic measures and making sure that people were sort of as far away in the classrooms as they could be, to make sure that the teachers are the ones who move from classroom to classroom, not the students, that people eat at their own desk. I mean, Denmark, for example, has done a pretty good job at getting back to school. We haven't done that. I feel the same way about work. I think if we were really thoughtful about hygienic measures, we could go back to work much more easily, but we're just not good at it. And so therefore we're really the worst country in the world. We have 4% of the world's population, almost 25% of the world's deaths. It's hard to watch. So I don't, what bugs me a little bit here is that we keep looking to the vaccine as the magic solution. Well, we have the much more powerful phenomenon we have is hygienic measures. And I mean, you hear, Robert Redfield say, the head of the CDC, wear this mask. And Tony Fauci says, wear this mask. And then the most recent data, you probably saw this, is that if 95% of Americans wore masks, we would save about 80,000 lives over the next few months. And the thing is, we don't know who they are, right? We don't know who those 80,000 people are. And that's why it's much easier for us to do this. There's an old awful quote from Stalin, which is, one death is a tragedy, a million deaths is a statistic. I mean, somehow the numbers are so numbing that we're numbed by them. They tend to mean nothing over time.
So, so one other question that's asked is that, what do we know about reinfection? Should people who have already been infected by COVID-19, should they get the vaccine when it comes out?
Yes, they should. The good news is that when they're doing these trials, they're getting antibody titers on everybody that enters the trial, and not saying if you're antibody positive, you're still in. So therefore, we'll have data on people who are antibody positive and still got a vaccine. So yes, I think the worst case scenario is you'll just get a booster response to your initial infection.
So as we wrap up, I think, there are a couple of takeaway messages here. First, you've really helped us understand, the process by which the vaccines are being developed, and, the resources that are being put into this effort is really the main factor in how we're able to do this so quickly. But that effort doesn't necessarily compromise, the safety and the efficacy tests that are being done. I think a second point is, helping us understand how, the course of the infection progresses, how different therapeutics can play a role in that, and why it's so important to have these testing processes in place to ensure that we're actually finding out the answers of what works and what doesn't. And that's true, whether we're talking about drugs or whether we're talking about a vaccine. But I think the most important piece is really to put vaccines into context of these broader public health measures, that, no single one of these measures is the magic bullet, as you said, right? That, whether it's masks, whether it's distancing, whether it's a vaccine, whether it's, opening all our windows and getting good ventilation, they're all additive, and we're kind of in it for the long haul. So, as we close, I guess the last question that I'd ask you is, how do we get people to buy into this holistic effort to bring this virus into check?
Well, I think we have to provide some level of hope that if we do this the right way, that we can get there. And I really do think we can get there. I think there will be a vaccine. I think the vaccines will be certainly more effective than the 50% level. I think that that in combination with hygienic measures will allow us to get on top of this virus, because I do think it's going to be a so-called single serotype virus. I don't think it's going to be like flu that mutates every year. So I think, therefore, when you get your 1 or 2 doses of this vaccine, I think that probably will protect you for a couple of years. I mean, we do have data on human coronaviruses, studies that were done in the early 1990s, where people were then experimentally challenged with that same strain they were originally infected with. And the protection was good a year later. I have no reason to believe it can't be 2 or 3 years. So I think we can get on top of this. We just have to really care about it as a society. We need to care about the person who we're standing next to. When people say, I don't want to wear this mask because, it's my freedom not to wear a mask. It's not your freedom to catch and transmit a potentially fatal infection. Not at all. You have to care about your neighbor.
And I think it's all of us working together, right?
Exactly. We have to care about each other. I mean, there's one movie, I'll close with this, I guess, is that There's a movie by the Sarah Cole Productions called The Polio Crusade. And you should watch this movie because it'll make you cry. When you hear people, those voices from the 1950s, there's such a community spirit. You know, you think my polio is bad. You should see what happened to Joe over here. And maybe it was because we were just coming off the shared national tragedy of World War II, where we saw the polio vaccine, Polio Crusade as a shared tragedy. You saw it a moment with 9/11, where we all hugged each other and pulled ourselves together. We saw us all being one, being a whole. And I think that's what's not happening right now. It's just way too divisive. And if we just see ourselves as all being responsible for each other, because we are responsible for each other, I think that we'll get past this.
I think that's a great note to end on. Thank you, Paul, for joining us in the middle of your busy schedule to share your insights with us. Thanks to all of you for joining us and for your continuous support of the Franklin Institute. I think this moment, more than ever, just helps us realize how important it is for science to be part of our decision making and for our society, and we couldn't do it without you.